In this lecture, we will learn about gluconeogenesis. Gluconeogenesis is the synthesis of glucose from non-carbohydrate substances like proteins and fats. Brain and red blood cells are completely dependent on glucose as the energy resource. Liver can fulfill their glucose requirements only up to half an hour from its glycogen reserves. So it has to take an alternative pathway in order to sustain the body. The alternative pathway is the gluconeogenesis. This gluconeogenesis pathway of glucose synthesis occurs during fasting. Gluconeogenesis is responsible for 64% of the total glucose production in the first 22 hours of the fast. The non-carbohydrate precursors include lactate, pyruvate, carbon skeletons of most amino acids except lysine and leucine, as these two amino acids give rise to acetyl-CoA, and another compounds capable of forming oxaloacetate, as oxaloacetate is the key precursor in TCA cycle, which is the energy deriving pathway. Acetyl-CoA cannot be converted into oxaloacetate in animals, but in the case of plants, this conversion occurs through glyoxylate cycle, which we will discuss in some other video. Coming to the pathway, pyruvate gets converted to oxaloacetate in the presence of pyruvate carboxylase. This step takes in the ATP molecule. Oxaloacetate in the presence of phosphoenol pyruvate carboxykinase takes in the GTP molecule and forms phosphoenol pyruvate. Phosphoenol pyruvate takes in a water molecule in the presence of enolase enzyme, forms 2-phosphoglycerate. 2-phosphoglycerate in the presence of phosphoglycerate mutase forms 3-phosphoglycerate. 3-phosphoglycerate in the presence of phosphoglycerate kinase takes in the ATP molecule and forms 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase forms glyceraldehyde 3-phosphate, and here NADH is oxidized to NAD plus and a molecule of PI is released out. Glyceraldehyde 3-phosphate in the presence of triose phosphate isomerase forms dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate combine in the presence of aldolase enzyme and forms fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate takes in a water molecule and in the presence of fructose 1,6-bisphosphatase forms fructose 6-phosphate. Fructose 6-phosphate in the presence of phosphoglucoisomerase forms glucose 6-phosphate. Glucose 6-phosphate takes in a water molecule and in the presence of glucose 6-phosphatase in endoplasmic reticulum forms glucose. Glycolysis and gluconeogenesis. Glycolysis and gluconeogenesis are reciprocal to each other, as glycolysis involves the degradation of glucose and gluconeogenesis involves the formation of glucose. Here all the enzymes are same except 3 enzymes which involve the formation of oxaloacetate from pyruvate. In gluconeogenesis, this step is formed in the presence of pyruvate carboxylase, and the second step involves the formation of phosphoenol pyruvate from oxaloacetate in the presence of phosphoenol pyruvate carboxykinase, and the third step involves the formation of glucose from glucose 6-phosphate in the presence of the enzyme glucose 6-phosphatase. Except for these 3 enzymes, remaining enzymes are same in both glycolysis and gluconeogenesis, but with their different allosteric interactions and covalent modifications like phosphorylation. Gluconeogenesis inhibits glycolysis at pyruvate kinase level by both allosteric interaction and by phosphorylation. Phosphoenol pyruvate carboxykinase is stimulated by glucagon, glucocorticoids, and thyroid hormones and is inhibited by insulin. Thank you.